Hello friends, how are you? Hope all are fine. Welcome to Sam Biology class. The term electrophoresis describes the migration of a charged particle under the influence of an electric field. Many important biological molecules such as amino acids, peptides, proteins, nucleotides and nucleic acids possess ionizable groups and therefore at any given pH in a solution exist as a charged species either as a cation which is positively charged or as an anion which is negatively charged. Under the influence of the electric field, the charged particles will migrate either to the positively charged electrode known as anode or to the negatively charged electrode known as cathode based on the nature of the net charges. Upon suspension in an aqueous solvent, almost all particles such as red blood cells, bacteria and many biomolecules acquire either positive or negative charges. The acquisition of such charges depend on the nature of the particle or the molecule and also the solvent. For example, proteins have many ionizable carboxyl and amino groups on their surface. These charge groups determine the net charge density of the protein molecules that make them to move in an electric field in a direction and at a velocity dependent on the sign and the quantity of the net charge density. If molecules have net negative charge, they will migrate to the anode, and if they have net positive charge, they will migrate to the cathode. This net charge density, which is dependent upon chemical groups and their numbers present in the molecules, can also be modified by the nature of the solvent. If we will increase the acidity of the solvent, the molecules will tend to become more positive, and if we reduce the acidity, then molecules will become more negative. This figure illustrates the principle of electrophoresis. These are two oppositely charged electrodes, anode and cathode, which have been immersed in a solvent that contain two different molecules A and B. And this is the electric supply. In first example, A and B have net negative charge, so they will migrate to the anode. And if A and B have net positive charge, then they will migrate to the cathode. Now the question is, if A and B have same charge, then how they will behave in the electric field? The answer is that they might not migrate together because of differences in their molecular weight. In second example, assume that A and B have the same charge, but A is larger than B. In this case, because of its small size, B will migrate at a faster rate toward the anode as compared to A. In third example, A and B have identical size and shape, but B has only one unit of negative charge, whereas A has 3 units. Because the net charge density of A is greater than that of B, A will migrate at a faster rate to the anode than B. Similarly, if A and B will have identical size and shape and also identical net charge density, then they will migrate together toward the oppositely charged electrode. From these examples, we can conclude that the electrophoretic mobility of a charged particle is directly proportional to its net charge density and is inversely proportional to its size. Combining these two, we can say that one of the most important factors that determine electrophoretic mobility is the charge to mass ratio. Each charge to mass ratio. So the molecule which have higher charge to mass ratio will migrate at a faster rate in the electric field. Taken together, these differences are sufficient to ensure the differential migration of molecules when subjected to an electric field. This is the basis of electrophoresis. Based on this differential migration, molecules can be separated on the basis of their shape, size, and net charge. This equation explains the mathematical relationship between various factors that determine the electrophoretic mobility. In this equation, V is the velocity at which a charged particle migrates in an electric field, and delta E by D is the field strength, Q is the charge on the molecule, R represents the size, and eta is the coefficient of viscosity, and eta is the coefficient of viscosity. From this equation, it is clear that the velocity is directly proportional to the electric field strength and to the particle size. On the other hand, it is inversely proportional to the size of the particle and to the coefficient of viscosity of the suspension medium. When the viscosity of the medium increases, the frictional resistance that acts against the movement of molecules also increases. There are three main factors that affect electrophoretic mobility of particles or molecules. Number one is charge. Number two, size, and number three, shape. The electrophoretic mobility of a molecule is directly proportional to its charge. So higher will be the charge of a molecule, greater will be its electrophoretic mobility. The charge, however, depends on the pH of the medium. Second factor is size. The bigger the molecule, the greater is the frictional and electrostatic force acted upon them by the suspension medium. Consequently, bigger molecules have smaller electrophoretic mobility as compared to small molecules. Third factor is shape. When migrating in an electric field, spherical particles will experience less frictional and electrostatic retardation, but the elongated molecules experience greater frictional resistance. Comparing globular with fibrous proteins, even if they have the same size, spherical particles have a greater electrophoretic mobility as compared to the fibrous proteins. Friends, this is just an introduction of electrophoresis. In coming days, we will be uploading many videos describing the principle, procedure and application of different types of electrophoresis. Goodbye and good night.